is nucleic acid sequencing. Typically, mainly DNA sequencing, but you can also start from the message. What is the message? The code is typically DNA, except people working on RNA viruses, that could be the code as well. What is the message? Do you know what I mean by the central dogma? What is the code typically for most organisms? DNA. What is the message? RNA. What is the functional part? It's protein, right? DNA replicates on itself to reproduce or others. And then when you have functional things, what is the next step would be from DNA? The message would be? The message is RNA. What is the process called? Transcription. Okay. And then what would be the functional thing is? We said translation is a process. Okay. Translation. And that gets into protein, right? Okay. So we'll agree on that. So, but let's focus on the DNA sequencing part. So, just to get you the historically, since the 1950s, we knew about DNA, whether we start with Rosalind Franklin, or Watson and Crick. We got into that, right, late 1950s. 75, we knew about bacteriophage genome. Sequencing pretty much started in the late 1970s. Maxam and Gilbert is a chemical degradation. Not so much used method. But then Fred Sanger, the Sanger method is still very commonly used, has been around at least since about the same time, in fact, little late 1970s. Here is what I just discussed. We discussed the central dogma, DNA transcribed into mRNA, messenger RNA, and the functional translation, right? But what we are trying to do in sequencing is, what is the order of these four bases? What is the genetic makeup of the organism you are working on? If does the A comes to next to G, next to C, next, the order of it is a key and high resolution question we deal with in sequencing. So there are different methods, various biochemical methods, but we will focus now mainly the basic Sanger dideoxy method. That is the key major sequencing method that enabled us and human genome sequencing, lots of it. There is a little older version, chemical degradation, we're not going to look at that, not much used. There is a recent version, a synthesis based on pyrosequencing. I have a different lecture, but in the interest of time, we're not going to cover it. It is on the online version, the pyrosequencing method. It's different, different from Sanger's dideoxy. It's a synthesis based. But you can understand that later. And then, the next generation, how do we do mass sequencing using Sanger, or the next generation? You definitely will look at the Sanger-based mass sequence as well. Okay? Again, what we will focus will be the Sanger dideoxy, which is also called chain termination method, which is the most common one used globally. So the Sanger method, the key elements, just similar to what you're using for amplification. You always need your template, genetic material, typically DNA. It might be from a cloned DNA fragment. You remember we use restriction digestion. You can clone it, attach it into a vector, transfect it into E. coli, and then you do sequence. Or it can start from a PCR product. A quick and dirty method, a little bit maybe, but you can have amplified fragment, amplicon, cut out of a gel, purified, run with sequencing. Well, it's possible. You need specific sequencing primers. There is the recombinant tag. Remember, I say here mutated tag is because you want to have a high fidelity, accurate, with a proofreading capability. Is very important. And then, very, very important, of course, your DNTP is important, but very, very important is your dideoxy. Okay, there is a typical deoxy nucleotides you use for amplification, but Sanger's method, the unique thing is the chain termination. We'll go through that in detail. For the chain termination purpose, you need a dideoxy NTPs. And we'll look at that. So these are the typical components that you have. Again, here is a dideoxy version of a base, just to give you an example. When you have a dideoxy attached, as you do your, it can be amplification, but for sequencing purpose, as amplify, and then you try to sequence. If you add, you have the typical components of the NTPs, right? Cytosine, guanine, thymine, and adenine. You also add a dideoxy. You can have different reactions, and technically it could be different, but in principle, you add a dideoxy NTP as a nucleotide as well, because if the dideoxy is attached at some stage, then Taq polymerase will fall, will not continue polymerizing your sequence. That's the key, the central piece of this principle of Sanger method. So each time, okay, in one well, in your reaction well, let's say this is a test tube, you will have all the 4 DNTPs, the normal ones. And then you add one of them, like the dideoxy ATP. Okay, I have the DDATP on that reaction. So polymerization continues. You run a regular, cycling. Polymerization continues until, at that, instead of the DATP, the dideoxy ATP attaches. When that attaches, Taq falls. No more polymerization. And by chance, of course, as the cycle continues, there will be a regular A attaching. You see A here, you see another A here, another A. There will be a short fragment, a longer fragment, a longer fragment, very long fragment, very, very long fragment, very, very, very long fragment. But each time the dideoxy attaches, Taq falls. Okay? The same holds true in your 4 reactions. The dideoxy TTP, the dideoxy CTP, the dideoxy GTP as well. So you will have a combination of 1 base pair difference, short and long fragments. Then you know which one comes first, so in order to order them and sequence your DNA. And if it's not clear, feel free to enter, but we'll look at the next few slides. We're looking at the same principle. So what is the process? You set up a PCR reaction, similar gene polymerization is the way we amplify things. You set up that polymerization similar to like we discussed in the PCR lecture. Once you run this, but this includes, the only difference is you have the dideoxy, right? And then you run your electrophoresis, and then you detect these various fragment combinations to order your sequences, to line them up. So